Hello students, today we are going to study an important topic which is the principles of cancer. In today's session, we are going to understand the definition, types of cancer, the properties of cancer and the pathogenesis of the disease. Before we understand the definition of cancer, we must understand the process in which a normal cell functions. When we talk about how a cell divides, we also talk about the cell cycle, which includes mitosis and the meiosis. The mitotic phase or end phase is followed by G1 or interphase, which is followed by S phase, in which growth and the DNA synthesis takes place. And finally, the G2 phase, in which final preparation for cell division takes place. In addition to this, there is also a G0 phase. A cell in G0 phase will not undergo any more division. Now, this process of cell division in a normal cell is regulated by two important processes. So you have the process of proliferation, which is responsible for growth, and the process of apoptosis, which is nothing but the programmed cell death, which helps to control the growth of the cells. So there are always a limit on the cell growth. However, when there is a balance disturbed between the process of proliferation and apoptosis, what we get is uncontrolled cell division. So cancer can thus be defined as uncontrolled growth and spread of cells. This cell is a representative image that you can see in the slide to show how cancer cells show uncontrolled growth and proliferation through the normal tissue. So basically, the term cancer is not a single disease, but it is a group of disease which is caused when abnormal cell divide rapidly and spread to other tissues and organs. Why exactly is cancer called a group of disorder? Because cancer can be different types. Depending upon the types, properties may vary. Though uncontrolled growth is seen in all metastatic cancer, the cancer could be different types based on sense of organ from where it is originated, the organ that is affected, and where the tumor is located or malignant. So let us understand the first type of cancer based on the cells of origin. The first type that we going to study is the classification is carcinoma. Carcinoma is the cancer of epithelial cells. An example here is basal cell cancer. Second type is the cancer of the tumor or tumor of the connective tissue, an example of which is osteosarcoma. The third type is the leukemia. So there is basically the cancer of blood forming cells. The example is chronic lymphoblastic leukemia. The next type is the lymphoma. These are the cancer of lymphatic system. This could be include tumors in tonsils, spleen and thymus. An example of which is Burkitt's lymphoma, which is an example of tumor or the cancer of lymphatic system. Next one is the myeloma. Myeloma are basically the cancer that originate in the plasma cells that circulate in the blood or form part of the blood. Example is multiple myeloma. So these are the types of cancer which are the cells from which they are originated. The next type of cancer is based on the type of organ that is affected. For example, the cancer that affect the lung is called lung cancer. The one that affect the breast is called the breast cancer. Similarly, the one which affects the brain is called brain cancer. Thyroid is called thyroid cancer. The next type of cancer is based on the degree of proliferation. Now, cancer can be of two types, that is benign and malignant. Benign means that is confined to a particular location. They do not spread, they do not invade other tissues, whereas in case of malignant, they are capable of invade surrounding tissues. so they are capable of spreading. They are the most lethal type of cancer. So far we have seen the definition of cancer, different types of cancer. So we have expand our knowledge to understand the properties of cancer cells and the pathogenesis of the particular disease. Now let us understand the properties of cancer cell. Cancer cell, they will show abnormal proliferation, differentiation, and they will see the process of apoptosis is suppressed. So they will show an increased lifespan or the survival as compared to the normal cells. Tissues, so now when we are going to understand the properties of cancer cells, we are going to understand that what we have seen in normal cell. Now, when we understand the properties of cancer cell, it is important for us to understand the different exists between normal cell and a cancer cell. So that the first important difference is that when you think about normal cells, they have density dependent cell proliferation. So what do you mean by density dependent cell proliferation? For example, if you have a cut or wound in your hand, your hands, in that case, there will be a growth of new cells which would help in the healing of the wound. So now this process of growth of cells will limit till the wound is healed. When the healing is completed, after that the cells won't grow or will not continue to differentiate. So that in normal cell there is always a limit as to how much the cells would grow. This means the growth is arrested once the cells reaches its limit. This process is not seen in case of cancer cells. They do not show a density dependent cell proliferation. They continue to grow, and so you would see a tumor formation or a lump formation which would take place. So another important difference in addition to this is what you observe is when you think about any cell to grow require growth factor. Now this growth factor requirement in every cell is defined. So it requires in some optimum amount, but when we think about cancer cells, this requirement is reduced, which means that very little availability of growth factor can also the cells can grow and proliferate. So because of that, it has the ability to proliferate or grow much more compared to that of the normal cells. Next important difference is that we observe is when we think about normal cell, they have something called cadherins on their surface. Now what is the cadherins? So these are the special type of proteins which give adhesive property to the cells, and because of this adhesive property, the cells will be able to maintain some resistance. They are unable to invade other cells. However, when we think about cancer cells, the cells there is absence of adhesive proteins, so they have the ability to invade and spread to other tissues. This cancer cell also have the ability to release something called protease, which is a protein digestive enzyme. So this enzyme digests the extracellular matrix of outer tissues, and because of the ability to invade, the ability to metastasis is much more as compared to normal tissue. Another important feature is that normal cells have different capacity of angiogenesis. Angiogenesis is the formation of blood vessels. They have a definite capacity. The cells grow and differentiate normally, and also they can undergo apoptosis, which is something called as the programmed cell death. It is after a certain lifespan or number of days they undergo programmed cell death, and so they have certain definite lifespan. Beyond the lifespan, they will not survive. This is known saving phase of cancer cell. So what we observe is that they have increased ability to angiogenesis, more number of blood vessels they can form, and because of that they can form differentiate. They tend to differentiate and undergo abnormal differentiation, and they fail to undergo apoptosis. That is, the programmed cell death is not seen in case of cancer cells, and there will be an increased lifespan in case of cancer cell. Now let us have look at how exactly this cancer differ from normal cell. So I said that they undergo differentiation, proliferation, and grow very rapidly. But if the way which they grow is not controlled, so you can see over here, that is variation in the size and shape of cancer cell if you compare to normal cell. Also there is nucleus is much more larger as compared to that of normal cell. They have abnormal number of chromosomes, and they form clusters without boundaries, so they are poorly differentiated. This is one of the important distinguishing factor which separate from normal cells, that is, even though they undergo differentiation, that the differentiation is not normal, they poorly differentiated the cells. Now, to understand the process of formation of cancer, we need to remember some important players in this process. So remember something called as the initiator, which initiate the process of mutation. Then something called as promoter, which promote the proliferation of the mutated cell. So remember one thing or two things rather than something. What is initiator, which is responsible for the mutation, and there is something called as promoter, which promote the growth of these mutated cells. Now what exactly is the initiator? So initiator is basically the agents that induces the mutation on the transformed cell. So initiation is going to the primary step, because unless the mutation is initiated, the cells will not show different properties than that of a normal cells. So that initiators are also called carcinogens. They are ones that induces the mutation. Let us understand the different types of carcinogens. They could be chemical carcinogen, for example benzene. They could be physical, or for example ultraviolet radiation. It could be infectious pathogens, for example Epstein-Barr virus or hepatitis B virus. Now this carcinogen tend to damage or destroy some genes, and by this process they induces mutation. Now there are some examples for carcinogens. There are many more. Apart from this, you just have to remember certain number, so that you know what is the basic classification and what are the some examples of carcinogen. Now once this particular cell which exposed to initiator or carcinogen, it undergo mutation. In that case they will undergo proliferation over a period of time. Now this particular cells which is exposed to a mutant or exposed to a carcinogen, called as carcinogen exposed cells or initiated cell. Now we can talk about the cells that undergo mutation. They are undergo initiation, and we talked about cancer cells and how they differ from normal cell. But at the cellular level, what type of carcinogens occur, what kind of mutation occur, we need to understand. That means, so with the help of this image, I will try to explain at the cellular level what kind of changes occur, what kind of mutation occur. We need to understand that as mean. So with the help of this image, I will try to explain at the cellular level, at the molecular level, what level of changes tends to takes place, because of which cell is undergone mutation. Differ some normal cell. So consider this is the image. In the top I have circled some cells which are the normal cells. If you observe this particular cells, you can see that there are a pair of chromosomes which are X chromosome, that is X1 and X2. There are two chromosomes. So even seen in some cases X1 is active and X2 is inactive. In some cases, some other cases, it is in opposite, that is X1 is inactive and X2 is active. So basically if you consider cells, there are mosaic of cells with different type of X. This basically mosaic of cells with different types of X chromosome being activated or inactivated. So this is how the normal structure basically would be. Let us understand now the whole process of how the cancer develops. So there is an initiator which comes into the picture first. Initiator, what are the initiator? What are the different types of initiator that we have already seen? Now it is the initiator will induce the mutation. So as you can see in the scene at the top of the image, we have number of cells, and the cells that has undergone the changes or the alterations is shown in the different colors. Now this cells which is genetically altered or which are undergo mutation will grow, proliferate in the presence of some promoters which is in the form of growth factors, which will undergo, as it grows it will undergo some additional mutations. Now in this process of undergoing mutations, what will happen is that the cells with this mutation will confer some advantages on the same. So like for example, if a cell undergoes mutation which will inhibit the process of apoptosis, then in that case that cell will not have defined span, which will continue to live for longer period of time. There is some kind of mutation because which the process of angiogenesis is increased. Then in that case this variability which will continue to grow and differentiate uncontrolled. So this some kind of mutation which will confer advantages on the cells, and which such type of mutation which confer advantages tend to become more dominant in this process over a period of time. Over a period of time, the cells which undergo such kind of selection mutation tend to emerge as more dominant cells, which is also for less clonal selection. Now what what will happen is this dominant cells will continue to grow which more rapidly. So you can see the cells over here which I have shown the pink color, but this pink color is also a mutated cell among this series or series of mutated cells which are shown in which in white color, orange color, but this particular pink color some same much more dominant cell is under on same mutations which have rendered it more dominant. These mutations have conformed some additional advantages on the same as you seen in the last image. As you can see in the last image, you see that this pink colored cell has grown and it is compared to that other mutated cells that it is become much more dominant, and the cells will now continue to multiply. And what you get, the end product is series of malignant cells which will now grow and invade other tissues, so that exact how the cancer cell emerge. So initially I repeat that you have initiator which will induce mutation. Now this mutated cell is called as initiated cell. This initiated cell will immediately not start growing. The presence of some promoter or some growing factor promoting oncogenes, this will proliferate or this grow once grows, it will have, what happens is growth is expanded or undergo series of mutation. So this mutation on conferring some advantages on particular cells. So for example, here initially you have this white stem. From this white cell with additional mutation, you have this orange colored cells which are emerging. These orange colored cells undergo some additional mutation, and you have this pink color cells which emerging. Now among this process, the cells among the process, the cell which undergo most selective or the kind of notations which conform advantages on the cells, like the stimulation of angiogenesis, inhibition of apoptosis. So the process confirms some advantages on the cell to continuously grow, proliferate, so that the particular cell emerges as the most dominant cells, which also called as a process of clonal selection. So ultimately how terminal selections are very invasive. Malignant cell emerges out. These cells has the ability to grow rapidly and invade other invasive tissues. This is because the most advantaged form of. Now let us just understand all the process with the help of an example. That is how the colon cancer develops. So that I told you earlier, there is something called as an initiator, because of which there is a mutation which is introduced. So now you can see that there is a year of cell there. There is a single cell which is mutated, which is shown in on it now. These cells, in the presence of some growth factors in that, now proliferated and has growth into number of cells. Now this particular cells continue to grow, proliferate. There is clonal selection, and this is clonal selection process. The most cell will be the mutations which are advantageous on the cell will continue to grow, become more dominant. So you have the process of growth of cell, where initially there is a formation of small and normal, which you can see now in these cells. These cells will continue to divide and form into intermediate adenoma. This will further result in the development of large idea. Finally, it is particularly say that there will be metastasis, that it will be tends to proliferate, invade other tissues, and main in this process invasion of the tissue is happening much more rapidly by invasion of blood and lymphatic vessels. So to blood and lymphatic vessels, it will spread to other tissues, and that is how the growth basically takes place. So that therefore So that is about today's session. In this session, we discuss the definition, causes, and the pathophysiology of cancer. Also, next we are going to discuss about the diagnosis of cancer, different diagnostic methods of cancer. This is the continuation of Neoplasia chapter. Here we are discussing about the various aspects of laboratory diagnosis of cancer. So basically in the next 10 to 15 minutes we will briefly discussing the various laboratory the methods utilized to diagnose cancer, and they utilize the management of cancer. So before we actually we talk about the specific method for the diagnosis of cancer, let us look some general considerations. So as soon as the patient comes with some various symptoms, the physician examines the patient thoroughly. So there will be a thorough physical examination to find some certain clues for the diagnosis of the cancer, and then the clinician advise some routine mystic things. Blood investigation probably will tell whether the patient is having anemia or polycythemia, which might be the secondary to the underlying malignancy. Okay, if it all is blood related malignancy, you can easily make it leukemia by examination of peripheral blood smear. Now let us talk about specific laboratory investigations in the diagnosis of cancer. The first and the foremost, cytopathological examination, where we actually study the cells, the diagnosis of cancer. And the first thing is that how are these cells obtained for the diagnosis. The two important methods for obtaining cells for the diagnosis, one is exfoliative cytology and the next one is an interventional cytology. In exfoliative cytology is basically the spontaneous exfoliated cells from the lining of the organism body cavities shedding. The examples for Pap smear would examine for your diagnosis of various lesions of cervix, and you can also examine these cells various body fluids like cardiac, cervix, and pleural fluid, etc. Right now, interventional cytology means that you obtain the material from the active intervention. What does it means? Example for interventional cytology finds needle aspiration cytology. So imagine the patient comes with the lumps in the breast, and you specify magnifies. So what the pathologist does is with the help of fine needle, you aspirate the material from the lesion and study the cells on glass slide. That's how you make a diagnosis of cancer by fine needle aspiration. Cytology. So next important one is histopathological examination, where the diagnosis is based on the microscopical features of neoplasm, where you can make accurate diagnosis in majority of cases. Now how are these tissues obtained? So the various methods are involved to obtain the tissues. One is needle biopsy. So in contrast to fine needle aspiration, this is a core needle biopsy. Needle consider the same example, the breast cancer. You take a small trunk of tissues by means of a cone needle. This is called cone needle biopsy, where you get material. Second one is endoscopic biopsy, particularly the GI magnifications. You know how the upper GI tract, which may include upper GI or lower GI endoscopy. to find out any lesions in the gastrointestinal tract and take the biopsy with the help of endoscopic procedure. The third one, which is incision biopsy, and this is a biopsy imaging. You have lesions, you are part of the lesions along with the normal tissues to study, particular to study this particular lesion is next one is the excision biopsy, where you knock out all the tumor along with the normal cells, uninvolved tissues to make diagnosis. So these are the various ways where you can one can get the material for diagnosis. So once you have made this probable diagnosis on the histopathological examination, you know that the next investigation is immunohistochemistry. Now what is this special investigation? Where it is used to detect specific proteins in these sections. How? By using antibodies that recognize and bind to those proteins, and also use a markers that allows visualization of this antigen antibody complex. So let us understand the more detail by example. Say for an example, I will take about breast cancer. Let us look into the determination of estrogen receptor on this cells of breast. Now why do you have look for the estrogen receptor? Because this particular patient or this particular region has the estrogen receptor on the cells of breast. It helps to predict the response of the cancer to hormone. So it is important to note whether the breast cancer cell is estrogen receptor positive or no. So let us take this example. So you take a section on glass. Let this small pink thing is a tissue section. Consider that the patient has estrogen receptor on this breast cancer cells. That is the target antigen, and you add antibody, and this antibody is called primary antibody, which can detect the estrogen receptor on the breast tumor cells. Right now there is an antigen antibody reaction. Now next you add the substrate, that is basically chromogen, which is substrate marker enzyme, which will be adding later. So this will be tagged the primary antibody. Now the substrate which is tagged to the primary antibody, and then you have the antigen antibody reaction provided. There is antigen is present. After that you add a secondary antibody. Now this antibody complex identify this complex. Alright, this secondary antibody is also tagged with an enzyme. So this is a peroxidase enzyme. This peroxidase enzyme, which is tagged to secondary antibody, oxidizes the chromogen to form a colored products. Okay, this is what you see under the microscope. These are the colored products where the nucleus is stained with brown color. This is how immunochemistry sites look up. Observe under microscope, and then you make diagnosis. Estrogen positive or the estrogen negative. If the estrogen receptors are not there, this brown color stain is not visualized on the nucleus. Right. So how the immunological chemistry helps? What are the applications of immunological chemistry? Based on they provide important information about the nature of the tumor, include its type and potential aggressiveness. It helps to differentiate between the different types of tumors, and also it helps to differentiate between different types of tumors, and also it helps in determining the extent of tumor involvement in the tissue. And finally, the most important one is the helping categorizing undifferentiated cancer by routine histopathological examination. You might notice able to make diagnosis when you do perform immunochemistry to find out exact diagnosis based on the antigen antibody reaction in immunochemistry. Now the next important investigation in the laboratory diagnosis of cancer is flow cytometry. Now, what is the flow cytometry? This is the technique to analyze the process of individual cells as they pass through the small nozzle in a fluid stream, and there is a light wave process through a stream of cells. You can scatter the light, and that inform the main passes through the stream. The cells now stack and the light, and then information is made use of this to be diagnosed various type of properties. So flow cytometry identification characterization of specific type of cancer, thereby you know flow cytometry support in this diagnosis and classification of cancer. Let me give an example. For example, leukemia. Looking at other in acute leukemia, some of this good leukemia often express specific surface. Example, CD10, CD19, and 20, which are and CD10, 20, which are not found in the normal cells. Okay, these are the proteins. These are the markers that are detected by using this method. Next method is the molecular techniques, where basically it means to that we are looking genetic makeup particular for this study. There are various techniques involved in this. The first one is polymerase chain reaction, which is to detect and amplify the DNA sequences associated with the cancer. Second one is fluorescence in situ hybridization. So in this method is used to detect genetic changes and chromosomal abnormalities. So third one is the gene sequencing, which used to identify mutation and other genetic changes. And the last but not least, the next generation sequencing. It is called NGS. This is the advanced method of reading DNA. The genetic changes in very short abnormalities of some case. These are some of the molecular techniques available in the diagnosis of cancer. Now let us look into the techniques. So examples of imaging technique are X-rays, where the low dose ionizing radiation. Next is the ultrasound, uses the high frequency sound waves. Next one is the computer tomography, so that is CT scan, uses X-ray and computer technology. Next one is the magnetic resonance imaging. So by use of magnetic field and radio waves, here the diagnosis takes place. Then there is a position emission tomography, that is the PET scan, where they are used to use a small radioactive material to produce detailed images. So these are the examples of various imaging techniques. These can be used either alone or common in diagnosis of cancer. So the next happening things is liquid biopsy. Now, what is the liquid biopsy? It is the technique where there is a use of blood, that is the liquid. You can detect a genetic makeup of cancer, and it is provides a real time snapshot of the patient cancer. Let me give an example. There is one of the tests called circulating tumor DNA test or ctDNA test, which detect the minute quantity of DNA which is shed by the cancer cell even before the symptoms of cancer are in the patient. Basically, it gives the information about the presence of cancer. Let me take an example of lung cancer. This lung cancer, even before magnifies it, patient might start shedding some two muscles in the might shedding some DNA material or even some proteins, and these proteins can be analyzed. You take a liquid, you can sample and subject this sample for analysis by molecular techniques. That what I told earlier is next generation sequencing, the revised transcriptase PCR. By means of this analysis, by means of identifying this no circulating DNA fragments, you can make out that the particular patient might be have this underlying cancer even before the cancer has manifested this patient. This is also used to monitor responses and detect the minimal residual diseases. Lastly, we are talking about tumor markers. These are the products of tumor now that can be detected in the cells or the blood or body fluids, which can be used to monitor the progression of the or the evaluation of disease or in case of cancer. For example, prostatic specific antigen, which is elevated in the prostatic cancer. There is a cancer antigen 125, so which is significant in ovarian cancer. There is another marker, that is alpha fetoprotein, which is increased in liver cancer. What is important to know about human markers is that lack sensitivity and specificity, because these can be elevated even in some non-neoplastic condition. So they should not be used. Presence of tumor markers alone should not be used. Diagnosis a might monitor cancer. They have to be combined with other examinations. Other examination like physical examination, radiational and other investigational method that earlier we discussed. Here we conclude the diagnosis of cancer. So that's all about today's diagnostic method. Next we are going to discuss about the grading and staging of the cancer. So we are discussing about the definition of grading and staging, different types of grading and staging and its examples. So what is meant by grading and staging? Grading and staging are basically, they are the methods or parameters which are used to quantify the probable clinical aggressiveness of a of a given neoplasm, which means they to say that where the given neoplasm you behaves aggressive or not. It is what we understand by knowing the grading and staging of cancer. So basically, they are used to evaluate the level of severity of cancer. Grading is basically on the degree of differentiation of the tumor cells. Differentiation basically means how close the tumor cells resembles to the normal tissue. Right, so grade is actually a measure of how abnormal this cancer cell look under the microscope. Right, so in contrast, stages is based on the size of primary lesion and the spread and it spreads, so it is the measure of how large the tumor is and how far the tumor has spread. Okay, so now you have understood the basic difference between the grading and stage, right? So grade is basically how abnormal the cancer look under the microscope, whereas in stage is basically how large the tumor is and how far it is spread now. Now there is a different categories of grades and stage. Grading is basically indicated by letter G, which is followed by a number quantified either low grade or high grade. If it is followed by a number, it is G1 to G4, G1, G2, G3 and G4, whereas the most common stage system use there will be whereas discussing in the great detail. Now it is back in 1920. You know this article from broadest division method to grade a sequence, grade muscle carcinoma into different grades. So based on that, most of the motorized most tumors, they are categorized into these grades: grade 1, grade 2, grade 3 and grade 4. Grade 1 means there are well defined tumors, which means the tumor cells look like normal cells or normal tissue. The cells, that why they are low grade. Tumor grade 3, where the most cells look abnormal and they are of high grade. So those tumors between two grades are called moderately different tumors. The tumor cells somewhat look abnormal and they are intermediate the grade categorized, whereas grade 4 is called undifferentiated tumor and the most of the cells, almost all the cells, right? So grade high grade, this is the high grade stage, anaplastic cells, and this grade is for highest. This is an example of squamous cell carcinoma. This is a well differentiated squamous cell carcinoma. You can make out the resembles to that of a normal squamous muscle in that evidence of keratinization. This is the squamous pairs, so it's what you seen in well differentiated squamous muscle carcinoma. Only few of these squamous spells are seen moderate differentiated carcinoma. In poorly differentiated carcinoma, we don't see any those, whereas undifferentiated carcinoma, it is actually difficult to make out this as square muscle. These are undifferentiated tumors. What we need to understand is grading. You can differentiate different kinds of carcinomas. What I have explained you is the squamous cell carcinoma. So grading can also be three types, about G1 to G4, right? Therefore that there is two tied, three tied means it is just differentiated, moderated and poorly differentiated, whereas two-third means you have low grade, high grade tumors. Okay, but what is important is as the grade increases, the tumor tends to grow and spread faster. That is very important. But the grading can be subjective. Not only that, it can be vary in different areas of same tumor. So that is a reason why stages is of graded value. Then grading in predicting the course of tumor. So let's see what stages is. We know that the stage is based on the size of the primary lesion and the spread, right? It is basically a measure of how large the tumor is and how far it is spread. See, most of the solid tumors, they are stated into 5 broad categories, which include stage 0, stage 1, stage 2, stage 3 and stage 4. So stage 0, there are no invasive cancer. There are carcinoma inside too, whereas in stage 1, which are early stage, where very small localized cancer. Say stage 2 is slightly larger, extended to nearby places stage. Stage 3 is much larger than the stage 2 and extended to nearby places and to lymph nodes as well as whereas stage 4 is the advanced stage, where the tumor has the spread into other part of the body. Now what help is staging. Now how actually staging, how stages neoplasm based on various parameters, like based on the physical examination finding, based on the laboratory investigation, could be routine your blood investigation or serum investigation based on a radiological examination, simple X-ray alternations or the CT scan or even scan position emission tomography scans, or finally it can also be this biopsy can also helps to staging of tumor. Incisional biopsy or the whole tumor can be assessed when the called excision biopsies. Now we are going to discuss about TNM classification. So the classification, which is developed by American Joint Committee on Cancer, it is also known as AJCC staging system, which helps in describing the cancer in great detail. TNMT stands for that describes the original or primary tumor. T for tumor, it describes the size induced of tumor, whereas N stands for describes the nodes, that describe the spread of cancer nearby nodes. M stand for metastasis, so basically it describes the metastasis are spread into distant organs or distant site. So there are different categories of stage. So I just explain only two differences categories. One is clinical staging and the another is pathological staging. We have discussed clinical staging where take help physical examination, laboratory investigation, radiological examination, biopsy, whereas in pathological staging include all these as well as complete pathological findings of excised tumor. So once the tumor is excised completely, the pathologist will study the entire tumor grade detail, and that also include this gives more precise information than the clinical staging, right? So this is the basic difference between clinical and pathological stage. Let us understand the staging of cervical cancer. So this is the stage 1. The cancer is confined in just the cervix, and stage 2 disease is inward in the cervix as well as the extended beyond the cervix, but not to the lower third of vagina. It's not extended too much beyond the upper 2/3 of the vagina, nor it extended into the pelvic mold. This is the stage 2. Whereas in stage 3, disease is extended into the pelvis as well as into the lower third of vagina. Whereas stage 4, as I told you, it inward into the distant organ. It can has inward as you know, rectum, bleed, bladder, and even the intestine even can be include. Distant metastasis to abnormal organism like liver and spleen. So that is stage 4. Now why do we need to know the stages? Staging conveys to the clinician. So basically we should know that the stages increase the 5 years of survival, that decreases the 5-year survival rate is drastically decreases. Look at the stage is on 5 survival rate is around 86%, whereas 4 and 5 is year survival is 7%. So that is the importance of stages of cancers. Now we discuss the grading and